It's situated in the Cappadocia region of Turkey. We just started working on CRISPR, and we haven't done so much. We are just beginners in our university. And here I will talk about different technologies available in CRISPR for plants, and some of the works that I have been doing at James Hutton Institute in the UK, and also in my university where I'm doing my PhD. While doing CRISPR-Cas technology in James Hutton, I was involved in doing knockout of potato. We are working with the potato plant, and with this, we were just designing the single-guided RNA, and transformation work is still going on because the potato work had this wild-type potato, which is a diploid variety, is little difficult to work with in case of transformation. This just simply shows how a viral adaptive immunity system from bacteria was transferred to the plants to be used. And there are 12 nucleotides, first 8 nucleotides which are resistant or tolerant to the proper targeting of the gene, and the last nucleotides, 12 nucleotides near to the PAM sequence, they are it should be specific. So some of the nucleotides that can go with the off-target genes are the situations which we often encounter in our research. So I just kept this slide for that. And in James Hutton Institute, my work was to mutate this dwarf-1 diminutor gene. This was basically involved in brasin steroid pathways of the potato, and mutating this gene, we were supposed to see the dwarf plants with reduced fertility in potato plants. And I was working with B91 plants, which was a diploid species. And here we used two different kind of tRNAs, because this gRNA which we designed was not having G nucleotide for that U6 promoter for that RNA transcription. So we used tRNA which has that G nucleotide, and we did multiplex sgRNA design. We cloned it into a vector which was designed by Abdullah Baraketi in Scotland. And these two guide RNA which are here targeted, one was dwarf-1 diminutor gene and the other one was the homolog gene. This homolog gene is very much homologous to dwarf-1, so the principal investigator Chaba, he wanted to mutate both of them so that this gene will not show the effect of dwarf-1 diminutor gene. So he wants to compare whether both of them get mutated, one, and the other one was dwarf-1 and homolog alone. So with lots of, and here we designed dwarf-1 diminutor single-guided RNA. We did the sequencing of the two genes, homolog gene of dwarf-1 diminutor as well as the dwarf-1 diminutor gene, and we identified that there is just one allele of this gene, and we designed a gene that targets dwarf-1 diminutor with a single-guided RNA in the PAM binding domain of the gene, and the same thing we did for the homolog. And in my PhD, I am working on cell wall invertase inhibitor gene, invertase inhibitor gene, which actually is supposed to increase the cell wall invertase gene, which is also known as apoplastic invertase or vacuolar invertase. Apoplastic invertase is associated with pathogenic resistance. So my hypothesis that if we could get to knock out this invertase, apoplastic invertase inhibitor, then naturally cell wall invertase enzyme will also increase, and then the resistance of the plants. And so far we haven't done so much. We have just cloned M6 variety, which is a diploid, and Desiree, which is a tetraploid, and we cloned around 10 clones of M6 and 17 clones from Desiree. From 10 clones of M6, we identified that there is just one allele, and with Desiree, we identified two alleles. And with the help of SSC, Broad Institute, and Scorer2, we found the different scores for sgRNAs. We identified the two highest sgRNA that is applicable. There is also one software, CRISPR-P, but it also shows that, like for off-target analysis, the same gene was also shown, so that sgRNA score was significantly decreased. So we didn't use that software. Instead, we used the software that is used by like other researchers also. And now, since we haven't done so much, we want to talk about some other technologies that have been done. One was Cas9 nickase, which was done by Foster et al. There was they had a mutation in asparagine to alanine in this Cas9 and nuclease, which creates a single nickase, and they identified that nickase doesn't induce non-homologous enjoin-mediated target immunogenesis, but they have homologous recombination, which is in similar level with the Cas9 nuclease. And in paired nucleases in rice, they have done this to reduce the off-target mutation with the help of Cas9 paired nuclease, because this Cas9 paired nickase is able to induce only one strand break, so two different targets at a time. Creating double strand break means more specific, so reduced off-target mutation. And with the help of inactive dead Cas9, Bortesi and Fusser, this is a review paper. It says that we can use it for transcriptional repression domain, for activation, to detect the sequence of the gene within a cell with the help of GFP, or for epigenetic studies using DNA methylase. And to reduce the off-target mutation, two FOC1 enzymes can be used, which is non-specific, and two dead Cas9, which cannot cleave the DNA, will be used to cleave the DNA. And to cleave the DNA, FOC1 enzymes will be used, so this will be more specific without off-target mutation. And in James Houghton Institute, they are also working on ribozyme with ribozyme method, where different promoters can be used. Like conventionally, U6 promoter is constitutional promoter, so constitutive promoters cannot be used for tissues specific cells. So tissue specific and other specific promoters can be used with the help of ribozyme, and they are working with this, and probably we will collaborate with them to work on this as well. And CRISPR-Cas12, which is also known as CPF1. Just now, James Houghton Institute, they told us that they had received this vector, and they will send us. So I'm working right now on CRISPR-Cas9, so we are also trying to compare CPF1 and Cas9 in the near future with our other students in my university. And with CPF1, because there is this is a T-rich protospacer. The gene which I'm working is highly T-rich. So working with Cas9, we found very few candidate single-guided RNA. So I think Cas12 could be a better choice for us to work with, because other researchers, it shows that this also induces a huge number of nucleotide insertions as well as deletions. So better option than Cas9 for us. And so that is what we are going to plan to work on in the near future with the help of Cas12 or CPF1. And this is the other technology which has been used in rice, programmable base editing in rice by Lu and Zhu. And here they have mutated T into Z nucleotide in NRT 1.1, which encodes for nuclear nitrogen transporter, and this replacement enhances efficiency in rice. And SLR1 that encodes for DELA protein with nucleotide amino acid substitution reduce height. And CRISPR-Cas13 could also be our near future working because my professor at the Alabash colleague is also working on Cas13, so probably we can collaborate with him to work on the other different of our gene of interest. And CRISPR-Cas13A, this is the other technology that has been done in like tobacco, where they have mutated RNA viruses and TMV viruses to reduce this, and it has been done only in protoplast, not in stable transformation work. And CRISPR-Cas13B, though this hasn't been worked on plants, there are different review articles that says that this can be also be worked on with plants in the near future. And there is one more technology which was recently published, self-elimination of CRISPR-Cas9 construct, where they use this suicidal gene, transgene in rice, where the pollens are directly eliminated if it has Cas9 gene, and if it's transgene free only, it will survive. So this can be used in offsetting the time that is required for selection of G crops that is free of transgenes without the use of backcrossing or self-crossing. And in conclusion, the first one which I'm working on is CRISPR-Cas9, and CPF1, which we will be working in the future, and probably the Cas13. These technologies we may be working in the future. So there should be, in conclusion, suitable selection of highly specific CRISPR technology that is required. Like for our case, probably CPF1 is better than Cas9, which we will figure out in the near future. And that's all about. Like when I was working with in James Hart Institute, I was collaborating with Adela Baraketi, who was the person who designed this vector, and Csaba Hornik was my principal investigator supervisor who was working on dwarf fungi gene. And in Turkey, my supervisor is Mehmet Emin Çalışkan. He's a potato breeder, famous potato breeder in potato in Turkey. And Alabax, who is here with me, is a famous person in transformation of potato. Thank you so much.
Thank you very much. Anybody has questions? Yes, please.
Thank you.
Hold on a second.
Thank you. My question is, you consider a lot of different tools, but as far as I understand, your aim is to create a simple knockout. So, and conventional Cas9 is sufficient to do it. So, for what application do you consider to apply such a big amount of tools?
Actually, for my gene of interest, which is cell wall inverses inhibitor gene, there is highly AT rich, and we don't know how CRISPR-Cas9 will work with that one because G and C content is very low, even less than 30%. So we thought that with CPF1, it can create more knockouts and more efficiently, because single-guided RNA option was very less in our case. And for CPF1, there is a huge option, so we thought maybe we can work with CPF1 in the future.
Thank you for your talk. First of all, I would like to say that I saw one statement in your presentation that CRISPR off-target effects are very rare in plants. I'm really scared about this statement. You know, we don't know very much about off-target effects in plants at all. Even human, you know, the recent publication was published only in June. And a second reason why I'm scared about it, because plant genomes, you know, are not very well sequenced at all. and a lot of sequences can actually functional, I believe, but we do not even input them or assemble them very well. And my question, actually, maybe I missed some information in this during your presentation. And what was the efficiency of genome editing in case of tetraploid potato? So how many clones you were able to isolate with 4 edited alleles?
We haven't edited yet.
Not yet.
Yeah, not yet. We just sequence the alleles of the gene yet, so far. Yeah.
Thank you.
More questions? I have a single one. So, how do you transform potato? How are you going to deliver your CRISPR components?
First, we will try with Agrobacterium. There is a deployed potato M6. This is very recalcitrant to transformation, and probably like James Huttenberg was also working with this, and they haven't been transformed yet, this M6 plants. And also there were some there were some scientists in United States, USDA, they were also not able to transform this. So probably for deploy, we may go to this gene bombardment. But if we can do with Agrobacterium in our institute, we will go with that. But with deploy, with tetraploid, we will go with Agrobacterium.
And so is it possible to regenerate potato from protoplasts?
We are not going to work with the protoplasts. Protoplasts?
Protoplasts.
We will not work with the protoplasts right now. And yes, they tried with the protoplasts also, I think.
Thank you very much. So I think if there are no more questions, then we thank you, our presenter.